In this lecture, we'll look at how genes are expressed. Through various experiments, it was determined that most genes in an organism's genome code for proteins. Here's the nucleotide sequence for a portion of a gene. Within this sequence, groups of 3 nucleotides correspond to a specific amino acid. A very specific amino acid. As you can see, the first 3 nucleotides, cytosine, thymine, and thymine, correspond to the amino acid glutamic acid, also known as glutamate. The next 3 nucleotides correspond to the amino acid serine. Then we have leucine, another glutamic acid, proline, and alanine. When these amino acids are chemically bound to each other, what kind of biological molecule is produced? Yes, a protein! The specific nucleotide sequence in a gene is the recipe for the sequence of amino acid that makes a certain protein. The central dogma describes the flow of genetic information in cells. Genetic information is stored in the DNA, in the form of a nucleotide sequence found in a gene. When the product of a gene is needed by a cell, a copy is made of the information in the form of RNA. The copied information is read by a ribosome, and a protein is produced. When a copy of a gene's DNA is made with RNA nucleotides, this process is called transcription. Then, when the RNA travels to the cytoplasm and the protein is made, the process is called translation. Before we examine the details of transcription and translation, let's answer a question. What happens when nucleotide sequences change? Let's look at the protein hemoglobin. Hemoglobin is found in the red blood cells of vertebrates. It is a quaternary protein and consists of 4 strands of polypeptides. These strands, or subunits, must come together to form the larger complex in order to function. Over 200 million molecules of hemoglobin are found in each red blood cell, constantly picking up oxygen in the lungs and delivering it to the cells. However, mutation in just one of the nucleotides coding for hemoglobin can result in a big change. On chromosome 11 of humans, we find the gene for one of the protein subunits of hemoglobin. The first 18 nucleotides that will translate into amino acids are shown and correspond to the 6 amino acids valine, histidine, leucine, threonine, proline, and glutamic acid, also known as glutamate. A genetic disorder that results in sickle hemoglobin causes the hemoglobin molecules to clump together, distorting the red blood cell into a sickle shape. These red blood cells become trapped in narrow capillaries, resulting in organ failure and eventually death. So what happened to the gene to result in hemoglobin that doesn't function like it should? The answer? One change, just one nucleotide. If a mutation results in the 17th nucleotide, changing it from an adenine to a thymine, then this changes the amino acid that will be translated. Instead of glutamic acid, valine will be the 6th amino acid, and valine doesn't act like glutamic acid, so it causes the hemoglobin protein to function differently and causes all those problems. Let's have a brief overview of the 2 major steps of protein synthesis: transcription and translation. Transcription is when a copy of the gene is made. The DNA is used as a template to produce the messenger RNA, or mRNA, by an enzyme called RNA polymerase. Translation is when the messenger RNA binds to a ribosome in the cytoplasm, and another type of RNA called transfer RNA brings the correct amino acids to build the protein. Remember that RNA is different from DNA in several ways. It is single-stranded, has ribose as its pentose sugar, and does not contain the base thymine. Instead of thymine, it uses uracil, which base pairs with adenine. Now, the language of genes is written in what is called the genetic code. Messenger RNA's nucleotides are a copy of the DNA sequence. Every 3 nucleotides form a triplet or codon, and each triplet corresponds to a certain amino acid. As we can have one of 4 different RNA nucleotides in each spot of the triplet, there are 64 different possible combinations. 61 of these code for amino acids, while 3 are signals to stop translation. Here's a table of the genetic code. Let's find out how to read it. Say you need to find out which amino acid has the codon UCG. As the first base is uracil, we would look at the left-hand side, the very first row. The second base is cytosine, so we would look at the second column at the top. The third base is guanine, so we would look at the fourth codon in that cell. So what amino acid does this codon correspond to? Yes, serine. You might notice that more than one codon corresponds to serine. Not only these four, but also these two. We say the genetic code is redundant because most amino acids have more than one codon that codes for it. This makes sense, as there are only 20 different types of amino acids found in living organisms. And here are the three stop codons, which serve as a signal to end translation. In addition, AUG is typically the first amino acid translated, and this codon serves as the start codon. Therefore, the initial polypeptide translated will start with methionine. Don't be confused if you see methionine somewhere else in the protein, however. It is not only the start codon, but also a regular codon as well. Here we have a hypothetical gene. You should be able to determine which strand is the template strand. Why? Well, RNA polymerase acts like DNA polymerase, as it must add nucleotides in a 5' to 3' direction. So which strand could be used to build an RNA strand that begins with AUG? Yes, this is our template strand. Now synthesize the messenger RNA. Again, you can pause if you need more time. Good job! So how many codons are in this sequence? Yes, 6. Now write down the messenger RNA sequence as you're about to see the genetic code table. Okay, here it is. What amino acids will be in your protein? Yes, methionine, alanine, asparagine, aspartic acid, and proline. Notice that even though you have 6 codons, one serves as a stop codon, so the actual protein is only composed of 5 amino acids. Prokaryotes have one type of RNA polymerase. It consists of several subunits, including the sigma subunit. The sigma subunit is very important as it allows RNA polymerase to bind to the correct location on the DNA template strand. RNA polymerase plus its sigma subunit is often referred to as the holoenzyme. Now let's look at the details of transcription. We will concentrate on prokaryotes, but eukaryotic transcription is very similar. The +1 denotes the start of the gene. Upstream of the +1 site is the promoter. The promoter has certain nucleotide sequences that the transcription molecules will recognize. Initiation of transcription begins when the RNA polymerase binds to the promoter region. A portion of the RNA polymerase called the sigma subunit recognizes certain nucleotide sequences in the -10 and -35 regions, forming hydrogen bonds with these nucleotides. The sigma subunit will open up the DNA helix. Only one DNA strand will be used to make the messenger RNA. This strand is called the template strand. After opening the helix, the sigma subunit has done its job and leaves. Now RNA polymerase can do its job and synthesize messenger RNA. RNA polymerase starts adding RNA nucleotides at the +1 site. It reads the DNA template strand in a 3' to 5' direction, but like all nucleic acids, the messenger RNA is built in a 5' to 3' direction, anti-parallel to the template strand. And RNA polymerase keeps adding RNA nucleotides until a termination signal is reached on the DNA template strand. The RNA nucleotides that are added to the messenger RNA have a specific sequence, which pair bond with their neighbors, resulting in a loop at the end of the messenger RNA. This loop causes the messenger RNA to separate from the RNA polymerase. This is only one type of termination signal, and there are quite a few others. And here's a detailed image of that hairpin loop with the messenger RNA molecule folded. Notice the RNA nucleotides base pairing with their neighbors. Now let's look at how prokaryotic and eukaryotic transcription differ. RNA polymerase in eukaryotes is also composed of different subunits. In addition, while prokaryotes have just one type of RNA polymerase, eukaryotes have several. RNA polymerase I transcribes genes that will not result in a protein product. Instead, the RNA produced by RNA polymerase I will directly be incorporated into the large subunit of the ribosome. RNA polymerase II does transcribe genes that code for proteins, and we will look at this one in more detail later. RNA polymerase III also transcribes non-coding genes, like RNA polymerase I. Okay, so other than using different RNA polymerases, how will the initiation of transcription occur differently in prokaryotes versus eukaryotes? Well, the sigma subunit plus RNA polymerase bind to the promoter in prokaryotes like bacteria. In eukaryotes, many proteins called basal transcription factors bind to the promoter. After this, the RNA polymerase binds. But wait, there's more. In bacteria, transcription and translation occur simultaneously because there is no nucleus separating the DNA and ribosomes. Also, the RNA transcript does not need to be processed in order to make the long journey out of the nucleus. This is not the case for eukaryotic transcripts. The RNA that is initially transcribed in eukaryotes is called a primary transcript. It is not mature messenger RNA because while it contains exons, sections that code for product, it also contains introns that must be removed. How are these introns removed? The introns must be removed and the exons spliced together. Protein and RNA complexes called small nuclear ribonucleoproteins, or thankfully SNRPs, form a complex called a spliceosome to accomplish this. First, the SNRPs bind to the nucleotides right where the exon ends and the intron begins, which is marked by the bases guanine and uracil. They also bind to an adenine near the end of the exon. More SNRPs bind to form the spliceosome, which pulls the intron-exon boundary close to that adenine near the end of the intron. This forms a loop in the intron. The loop is cut out, and a phosphodiester bond is formed to link the exons together. After all the introns are removed from the transcript, a continuous coding sequence is formed. In addition to the removal of introns, the transcript is processed some more. Remember when RNA polymerase was making the transcript? Well, a cap is immediately added to the 5' end. This cap consists of a molecule called 7-methylguanylate and 3 phosphate groups. And after the transcript detaches from RNA polymerase, a tail is added to the 3' end. This tail consists of over 100 adenine nucleotides. Now, neither the 5' cap nor the poly A tail are translated. So what good are they? Well, the cap and tail prevent the messenger RNA from being broken down by enzymes in the cell. In addition, the cap and tail are recognized by the molecules of translation, allowing the messenger RNA to bind to the ribosome. Other parts of the transcript, called untranslated regions, or UTRs, help to make the messenger RNA more stable and regulate events during translation. Then after the eukaryotic primary transcript is fully processed, it will leave the nucleus.